Okay, well, thanks for the introduction, Lin. Today I'm going to present some of my research on quantum susceptibility MRI for imaging brain during aging and neurodegeneration. Okay, the next slide. So in my today's presentation, I will give some basic introduction about magnetic susceptibility and how we can use it as an intrinsic tissue contrast for imaging the brain. And then I will talk a bit about quantitative susceptibility mapping and how we can use it to imaging brain iron and myelin content during aging and neurodegeneration. And at the end, I will talk a little bit about susceptibility anisotropy and susceptibility tensor imaging. So what is magnetic susceptibility? Magnetic susceptibility is a dimensionless proportionality constant that determines the degree of magnetization of a material in response to an applied magnetic field. This is according to the definition in Wikipedia. Of course, you probably have seen this in the very basic physics books, which defines the susceptibility in the relationship between the magnetic flux density B and the magnetic field strength H, with the relative magnetic permeability being just 1 plus chi. Chi is the magnetic susceptibility. And according to the magnetic susceptibilities, different materials can be categorized as either diamagnetic, which means their susceptibility is less than 0, or when the magnetic susceptibility is larger than 0, it can be either paramagnetic or ferromagnetic. And of course, materials with different magnetic susceptibility have different behaviors when they are placed in the magnetic field. For the diamagnetic materials, usually in its natural form, it does not have any net magnetic moment in the molecular level. And if you put it in the external magnetic field, it will induce some local magnetization that's opposing the external magnetic field. And when you are taking the material out of the magnetic field, it will return to its natural form. And for paramagnetic materials, usually it has some net magnetic moment in its natural molecular form, but they are generally oriented randomly. And when you place them into an external magnetic field, those net magnetic moment will align with the external magnetic field, thus creating some intrinsic magnetization enhancement for the local magnetic field. And when you place them out of the magnetic field, they will also return to its natural form. And for ferromagnetic materials, they have some small domains, they call magnetic domains, which have kind of aligned magnetic moment. And if you put this kind of material in an external magnetic field, all these net magnetization will align with the external magnetic field, creating an enhancement in the local magnetic field. And if you put them out of the magnetic field, they may also have some kind of memory to have this magnetization, this magnetic moment still aligned. And that's actually how we make those permanent magnets. And in terms of molecular mechanism, the paramagnetism mainly comes from those unpaired electrons. There are some kind of magnetic moments generated by the nucleus, but it's usually negligible as compared to those generated from the electrons. And it's also well known that the paramagnetism depends on temperature according to the Curie's law. So the higher temperature, usually the lower the magnetic susceptibility. And for diamagnetism, usually those materials does not have unpaired electrons. And the diamagnetism usually comes from those the tendency of those moving electric charges trying to shield the interior of the material against the external magnetic field. So, and also the diamagnetism does not depend on temperature, but according to the molecular itself, it may have some kind of anisotropy arises from the asymmetry of the molecular bonds. So this slide shows a kind of full magnetic susceptibility spectrum. So on the left, you have these kind of diamagnetic materials, and an interesting phenomenon that you can see here is like for these diamagnetic materials, it can create certain kind of diamagnetic levitation, which means the induced internal magnetic field opposing the external magnetic field is strong enough to counterbalance the gravity. So that's an interesting physical phenomenon. And also in the middle, we have the paramagnetic materials, and on the far right, we have the ferromagnetic materials, which means their magnetic susceptibility is very strong. And you usually see those kind of materials probably in your MRI safety trainings. And when we talk about biologic tissues, because most of the biologic tissues are mainly composed of water, so the tissue magnetic susceptibility is usually around the water magnetic susceptibility, which is -9 ppm. And according to their tissue compositions, it may have slightly more paramagnetic or slightly more diamagnetic as compared to water. For example, as we know, the deoxyhemoglobin and also like the liver with highly iron overload would have a more paramagnetic property than water. And when we look at the brain, as far as we know now, the major paramagnetic sources in the brain is iron, either in its ferric or ferrous form. And about 2/3 of the irons stored in our body is actually bound to the hemoglobin in our blood pool. And of course, its magnetic susceptibility depends on its oxygenation levels, and that's actually the well-known mechanism for the Bode effect. And another one sort of the iron source is actually in its non-heme form. So those are irons in the cell. And among them, only a very small percentage, like less than 3%, are actually actively involved in the cellular biochemistry. And about 97% of those irons are actually stored in the ferritin cage, which is kind of a spherical proteins that can store a lot of iron atoms inside the protein. And also there are other forms of tissue irons like the neuromelanin or hemosiderin, etc. And there are also diamagnetic sources in the brain, and the major diamagnetic in the brain is the myelin, and mainly in the white matter. And one thing I want to point out is due to the highly organized microstructure in the white matter, the magnetic susceptibility of myelin, she's actually shows some bulk anisotropy, as we will talk more later. And there are also other sources of diamagnetic tissues like calcium or proteins. But in normal conditions, those sources are usually either at very low concentration or very weak diamagnetism. So what does the magnetic susceptibility do in MRI? So we know that in the in MRI, different tissue magnetic susceptibility will cause kind of a local magnetic field shift, and thus of the lambda frequency shift. And such kind of spatial variation, it was usually considered as a kind of source of susceptibility artifacts that may cause signal loss or image distortion. But from another point of view, this kind of susceptibility variation can also be viewed as a kind of intrinsic tissue contrast. So as shown in one of those early studies from the NIH group, they have shown that the susceptibility related tissue contrast can actually help reveal some very important anatomical structures, such as the cortical layers. So in the last decades, the quantitative susceptibility mapping has been developed as a kind of imaging technique to reveal the underlying tissue magnetic susceptibility from the measured MR face data. And QSM solves this kind of QSM estimate the susceptibility maps by solving this kind of dipole deconvolution problem. So the relationship between the susceptibility source and its induced magnetic field change, or the phase measures, can be described as a kind of a dipole convolution formula. And by solving this kind of dipole deconvolution, we can then estimate the susceptibility sources. So it should be noted that in the QSM, we assume the tissue magnetic susceptibility is always isotropic. And for doing QSM, we usually use a kind of standard gradient echo sequence, usually multi-echo. And then we use the phase image to derive the QSM maps through multiple processing steps, including like phase unwrapping, echo combination, masking, and background field removal, and also finally the field to susceptibility inversion. So of course, you have probably seen in the literature there are many variations of methods for doing each step, but if you're interested, you can also go into some of the recent review papers, and our SMRM EMTP study group is also trying to get a consensus paper out since last year. So it's probably going to come out very soon. So that would be helpful to understand like each step and how to choose the acquisition and the processing parameters. So usually for doing QSM, there are like different probably challenges for doing each processing step, including like coil combination, unwrapping, and also background removal. So I'm not going to talk about those details, but there are some very good recent review papers talking about like different methods on how to solve these issues. But of course, one of the major challenge for doing QSM is the field to susceptibility dipole inversion. And such kind of, this is kind of like an equipost inverse problem. And the equiposteness actually comes from the singularity areas in the dipole kernel, which usually leads to the streaking artifact or like noise amplification if it's not handled correctly. And also because for the MRI measurement, we can only measure, get the phase measurement from MR visible regions. So we have to have like certain amount of proton density to get any signal. So this all leads to the equiposteness of the QSM dipole inversion. And of course, in order to solve this equipost in QSM inverse, we can either use a kind of a data oversampling strategy like used in the Cosmos approach. So in this method, we kind of acquire the MR phase measurement at 3 or more head orientations. In that way, we can actually feel the singularity regions in the dipole kernel, and then the inversion becomes a kind of overdetermined problem. Of course, this is the Cosmos is usually considered as a gold standard approach in QSM, but it's kind of expensive in terms of acquisition. In most of the clinical cases, we usually just acquire the phase data in one head orientation, and we'll need to use some kind of regularization approaches for the dipole deconvolution. And basically, the idea is we put the QSM inversion into a kind of optimization problem using a regularization term that can incorporate some kind of a priori information, either from like the morphology of the brain structures or some sparsity type information. Of course, the question you may want to ask is why we want to do QSM, given there are like many other MR techniques that are sensitive to tissue magnetic susceptibility, such as the T2 star weighted or the SWI susceptibility weighted imaging, or the phase maps or frequency maps. So as a matter of fact, QSM actually has a couple of advantages over its predecessors. So the QSM overcomes the blooming artifact, which is due to the non-local field change generated by a kind of neighboring susceptibility sources. And also the previous techniques usually have a stronger dependence on subject orientation and image acquisition parameters, which QSM has some advantage. And also in the QSM maps, it's easier to separate the paramagnetic source, such as iron, versus the diamagnetic source, such as the calcium. So here, as the arrows points, we can see there's some calcium depositions in the coronoid plugs in the ventricles, but they show like similar contrast as compared to the basal ganglia iron signals in the previous techniques, but we can see the difference in the QSM map. So due to the high signal to noise and contrast noise in the phase measurement, we can actually get a pretty high resolution QSM maps, especially at ultra high field. So I'm showing here just an example QSM images we acquired here at 7 Tesla with 0.5 millimeter isotropic resolution, and we can appreciate a lot of detailed anatomical structures. And if you can effectively correct the motion and like the field fluctuation related to physiological noise, such as breathing, using a kind of advanced navigator correction, for example, we can further push the resolution as shown in this work, this recent collaboration with John Liu in UT Southwestern. So here shows some example images of the T2 star weighted and QSM images acquired at 0.3 millimeter in plane resolution. And if you look closely, you can see there are some very detailed anatomical structures like the line of January or the superficial white matters, and also the details of the hippocampus. Of course, besides seeing the brain structures, we are also interested in measuring the tissue compositions and also brain functions in different neurological diseases. And because of the major paramagnetic sources in the brain is tissue iron, we can normally infer the tissue iron content from those positive QSM measurement. And over the years, there are several validation studies that have compared the QSM measures with histochemically measured iron content in the subcortical gray matters. These include like using mass spectroscopy or like X-ray emission, and also the classical power staining for irons. And all these studies have found pretty high correlations between the tissue susceptibility measured using QSM versus the irons measured using the other method. So, of course, we want to ask why we care about the irons. Actually, iron itself is a very important element that involves in a variety of biological mechanisms, including oxygen transport or neurotransmitter synthesis and myelin production. And the iron homeostasis is usually highly regulated in the body and also in our central nervous system to avoid its toxic effect. And also iron is known to be closely related to aging and neurodegeneration. So in our brain, actually, the irons will progressively accumulate while we get older. This has been like well documented, like in the early 1950s, and also confirmed later in like MRI measures using QSM. And during neurodegeneration, the iron homeostasis may be changed due to like inflammation or maybe change of BBB function. And iron overload actually has been reported in many different neurodegenerative diseases, like the Parkinson's disease or Huntington's disease, Alzheimer's disease, and multiple sclerosis, etc. And also iron deficiency or loss may also be involved in some neurological disorders. So I'm just going to show some examples of these kind of applications of QSM in neurodegeneration. The first example is in the Huntington's disease, which is a genetic disorder. And the genetic mutation in the CAG expansion can actually be detected many years before the symptom onset. And for brain changes, besides usually seen like brain atrophy, the iron overload in the striatal-cortical pathways is believed to be another kind of signature in this HD pathophysiology. Here, just showing an example of the QSM images in two subjects, one with Huntington's disease and another age-matched controls. So we can see there are a higher magnetic susceptibility in the deep gray matters, especially in the putamen and caudate. So far, we have done actually two separate studies in using QSM in the HD in the HD groups as compared to controls. So in both studies, we found like consistently higher magnetic susceptibility in the striatum and also the pallidum in the pre-manifest or early HD group as compared to controls. And more importantly, such kind of increased iron levels in the striatum were found significantly associated with the CAP score, which is the genetic mutation burden. So this kind of findings indicates the iron is likely part of the HD pathophysiology. And in a very recent kind of small longitudinal studies, we also found there's significantly higher iron deposition rate in the caudate and also globus pallidus in the close to onset and also early HD group as compared to controls. So this kind of indicates the disease progress of iron deposition may, as the disease progress, the iron deposition may get kind of worse and worse. So another example is the Alzheimer's disease, which is known as the most common cause of dementia. And also it's known that for Alzheimer's disease, the high amyloid beta deposition is the is the pathological signature of the disease, which is usually detected in like CSF by lumbar puncture or using PET imaging early in the disease. But on the other hand, it's also believed that the cerebral iron overload may play a role in the AD pathogenesis, as irons has been found kind of co-localized with the A beta plaques, like using autopsy. So there are some hypotheses saying that the interactions between iron and the beta amyloid may kind of further promote the cognitive decline in the AD patients. And of course, it should be noted that while the tissue iron is paramagnetic, the beta amyloid protein is actually weakly diamagnetic, as shown in some recent phantom and mouse studies. And of course, the hypothesis of the potential interaction between brain iron and the beta amyloid has been supported by some recent QSM and PET studies, which measures both the QSM iron using QSM and the beta amyloid using the PET. So this is one of the example study by an Australian group showing that the iron content or the QSM measures in the hippocampus is actually significantly associated with the subject's cognitive decline over time, but only in the beta amyloid positive group. While in the negative group, we don't see such kind of association. And here shows another study that we collaborated with Paul and Shaw in Zurich. So in this study, we show that in this cognitively normal subject, there are some brain regions, for example, in the temporal lobe, we see a kind of correlation between the A-beta measures and the iron measures. And the A-beta levels in these regions can actually better differentiate the cognitive functions of the subject groups. And in a very recent study we did here in an even younger population, these are like cognitive normal subjects. We found a kind of strong association between the subject's cognitive functions and also their cognitive function in certain domains like the memory or visuospatial functions. The association between the cognitive function and the magnetic susceptibility in the hippocampus. And this kind of association is even stronger in those A-beta negative groups, which seems to suggest that the tissue iron may have kind of contribution to the cognitive decline independent of beta amyloid at this early stage. And the interaction between iron and beta may happen in a later age. Here, just showing kind of another results we observed using the voxel-based analysis, showing kind of similar results of association between the cognition and magnetic susceptibility, kind of independent by beta at this early stage. So another important application of susceptibility MRI in neurodegeneration is in the multiple sclerosis, which is a autoimmune disease with demyelination happening in the central nervous system. And of course, there are many studies reporting the iron content may change in the subcortical gray matters in MS, but also in white matters, we may see both changes of tissue iron and myelin content. As shown in this histology images, we can see in the white matter, especially in the superficial white matter, we see kind of co-localization of myelin and iron. And those iron are likely stored in the oligodendrocytes that's forming the myelin. But of course, inside the MS lesions, we know that the iron deposition and demyelination are expected to increase the magnetic susceptibility, but they may have a slightly different effect on the R2 star measurement. So in one of our early studies, we look at the MS lesions. We do see quite some QSM hyperintense rim lesions. They show like higher susceptibility values, indicating kind of demyelination and also iron deposition. But on the other hand, we also see quite some MS lesions with QSM iso-intense contrast. And these lesions show up in the flare image, the T1 image, and also are hypointensity in the R2 star maps, but they appear kind of iso-intense in QSM. And of course, a reasonable hypothesis is that in these lesions, we see both demyelination and iron loss. But of course, it would be ideal if we can kind of tease apart the contributions from brain iron and myelin using the MRI contrast that we can see directly. So actually, the idea of QSM source separation has been in the field for many years, but were actively pursued in the last couple of years, leading to multiple papers. And the goal is trying to separate the paramagnetic and diamagnetic sources co-localized in a kind of MR imaging voxel. And the basic idea is that we can combine the information from QSM and R2 star or R2 prime to disentangle the contribution from the positive and negative susceptibility source. This is, of course, technically possible because the paramagnetic source and the diamagnetic source, they kind of contribute additively to the relaxation, but they contribute subtractively to the bulk magnetic susceptibility. Here I'm just showing one of the recent paper from the Korean group on the chi separation work. And so here the chi positive maps can be considered as a measure of iron content. The chi negative maps can be considered as a measure of myelin content. So interestingly, they also show these two types of MS lesions. So in the paramagnetic rim lesion, we do see like increased positive susceptibility and decreased negative susceptibility, indicating iron deposition and demyelination. But for the QSM iso-intense lesions, they have shown that in these kind of lesions, we do see a decrease in both iron and myelin content, which kind of agrees with our previous hypothesis. So this is very nice to see using the susceptibility measurement directly. So as a kind of interior summary, I have shown that QSM can give a pretty accurate and reliable mapping of the bulk tissue susceptibility, which can be a good measure of the brain iron content in gray matter. While the QSM source separation for paramagnetic and diamagnetic susceptibility components can further help quantify the co-localized tissue iron and myelin changes. And of course, there are other applications for QSM in the neurodegeneration, for example, in TBI, and also like it may help doing like surgical planning for deep brain stimulations because of the very nice contrast shown in the QSM maps. So of course, there are still some remaining technical challenges for QSM as summarized in our first QSM challenge. So we know that the Cosmos solution is the gold standard, but it's expensive in terms of acquisition. And for single orientation QSM, usually we need a pretty careful tuning of the hyperparameters. And if we tune it over certain metrics, you may get kind of over smooth QSM maps. And also we know in QSM, we used a simplified isotropic susceptibility model. So after the first QSM challenge, there's kind of a development trend for deep learning based QSM, like the work in the QSM net and also deep QSM. And for most of the deep learning based QSM, the basic idea is we train a powerful neural network that can approximate the kind of nonlinear dipole inversion functions to get the QSM maps from the local magnetic field. Of course, the QSM, the deep learning based QSM has a lot of advantages. It can create high quality QSM reconstruction with minimal artifacts and no over smoothing, especially when the network was trained with kind of the gold standard Cosmos labels, and the calculation is super fast. But there are also some cons for the deep learning-based QSM. It kind of suffers from a limited generalization capabilities, which means because the neural networks trained is highly kind of a data-driven approach, so its performance may kind of degrade if your test data is quite different than your training data set. And such kind of difference may come from like different acquisition resolutions, like noise levels or the susceptibility range, or even the acquisition orientations. So as a result of such kind of limitation, the deep learning-based QSM actually didn't outperform the conventional approaches in our second QSM challenge. And here you can see that the deep learning-based method represented by these green lines are actually having larger errors as compared to the blue lines, which represents the conventional iterative approach. So this leads to our kind of development on this LPCN QSM, which is of course a deep learning based approach, but we tried to have the deep learning network to take into account our known physical model of the dipole convolution for QSM. And this work was in collaboration with Jerry Myers and Kuo Wei in the biomedical engineering department. And so basically the idea is we only use the neural network as a kind of a regularizer and make it to learn some prior information that we commonly use in the conventional iterative optimization. And here just shows some example of the reconstruction results. As expected, the LPCN QSM can give kind of comparable or even better reconstruction as compared to conventional method and other deep learning QSM approach. And more importantly, these LPCN can be much more flexible to be to incorporate, for example, measurements of face images at different head orientations, or even we can have like a more face measurement to feed the neural network to give like even better QSM reconstruction. So as we talked about before, the QSM assumes like the isotropic magnetic susceptibility, which can be described as a scalar. In this case, we would expect the magnetization is the amplitude of the magnetization is orientation independent, and the magnetization would always align with the external magnetic field. But as we talked about in white matters, the magnetic susceptibility may be anisotropic, which means in those tissues, the magnetization may not always align with the external magnetic field, and the amplitude of the magnetization may be orientation dependent. So actually, such kind of anisotropy has long been observed in brain tissue. For example, as shown in one of these early studies from Chen Liu's group, they've shown that in the mouse brain, if we look at the white matter, the QSM measures become more diamagnetic when it's perpendicular to the field, and it's more paramagnetic when it's more kind of parallel to the field. And similar phenomenon has been consistently observed in humans. And here, like you can see different white matter contrast when we measure the QSM at different subject head orientations. And in terms of its molecular mechanisms, we have briefly discussed the susceptibility anisotropy actually in the white matter actually comes from the highly organized microstructure. And these microstructure are also composed of molecules with susceptibility anisotropy, such as the myelin lipid. So if without such kind of structures, the bulk susceptibility anisotropy would disappear. This was like nicely demonstrated in the early studies from Chen Lei's group in the chivalrous mice. So for the chivalrous mice, they don't have the myelin sheaths, and then you would see no or very negligible orientation dependence of the susceptibility. So such kind of anisotropic susceptibility would be better described using a tensor model. And if we use the tensor model, we can extend the QSM into the susceptibility tensor imaging. But of course, this is more complicated because for QSM, we only need to solve like a scalar for each voxel, but for susceptibility tensor, we need to solve 6 unknowns for each voxel. So theoretically, we will need more measurement of the phase. So for doing STI, it's kind of similar like doing QSM. So basically, we need to acquire more phase data from more head orientations. We do a kind of volume co-registration. We do similar phase pre-processing to remove the background, and then we can combine them to feed the susceptibility tensor. With the susceptibility tensor, of course, we can also calculate the tensor components of, like, for example, anisotropy, and also even do tractography like what we do using diffusion tensor image. So in the last decades, many works, mainly from Chen Lei's group, they've shown that the susceptibility tensor imaging can be very useful tools for MR histology, but this is mainly for ex vivo studies, like in the mouse model or some different tissue parts. But beyond the preclinical imaging, STI may be pretty challenging. So STI, of course, has the potential to probe the tissue microstructure. It may has the advantage to become very high resolution imaging of the microstructure. And it's also sensitive to different chemical components like the myelin instead of just the structure. But it also has a lot of challenges due to its complicated model and data processing, and also it requires the subject rotation, which can lead to longer scan time and the patient discomfort. And more importantly, it's kind of a ill-post inverse problem, especially when we have a limited orientation sampling using the normal MR head coil. Here, just an illustration of this kind of a challenge due to the limited orientation sampling. So for solving the STI inverse problem, ideally, we want to sample the orientation space in a wide range over space. Like, well, if you can rotate your brain like all, that would be ideal. But in practice, for most of the MR head coils, we have a limited space, which makes the STI inverse ill-post. So as a result, even if we can acquire a large number of head orientations, like the studies done by the MGH group using highly advanced acceleration with the wave Chi-P sequence, that we can acquire one GRE scan in less than 2 minutes, and they acquired like 15 head orientations. The reconstruction of the susceptibility tensor is still not ideal, and we can see that the susceptibility anisotropy usually looks pretty noisy, and the tractography results is not as reliable. So of course, we can also use some kind of regularization trying to get a better STI reconstruction. For example, we can regularize the anisotropy to be within the white matter. And also we can apply some morphological a priori in the STI inverse. Here just showing some example of the a priori information we may be able to use for the STI inversion. And as shown in our previous studies, such kind of regularization, of course, it's helpful. It can improve the STI reconstruction, giving like a more homogeneous susceptibility anisotropy, and also a better estimation of the fiber directions as compared to the DTI. And over the last few years, there are different approaches that have been proposed to further improve this. So here just showing the examples, we can of course using the mutual information from R2 star relaxation tensor, we can use the diffusion measurement to help improve the STI reconstruction. But of course, if you look at the results, they are still not as kind of good quality as compared to the diffusion measurement. So since last year, we have been working with Jerry and also Zhenghan on this deep learning based STI, which we call the deep STI approach. So this work is aims at to improve the STI reconstruction using a kind of data-driven priors that we can learn using a powerful neural network. And it's kind of similar like the LPCN QSM idea, but the deep STI, of course, it's kind of similar like LPCN QSM, so that we train the neural network as a regularizer that can be used to learn the a priori information for the STI inverse reconstruction. Of course, this is much more complicated than the QSM inverse because we have like at least 6 times larger in the variable space. So for this work, we use the training data that's generated from a simulation. Because it's quite challenging to obtain a high quality human STI in vivo. And we created this STI brain phantom based on available Cosmos data and diffusion measurement. And then we simulated the field change and used the simulated field change and the STI phantom as a training data pair for the network. Here just showing some results of the reconstruction using simulated data. As we can see, if we have, okay, here showing if we have like a large number of head orientations, conventional method and the deep STI method can give a fairly reasonable reconstruction with the best result given by deep STI. And if we have a smaller number of head orientations, the conventional method usually degrades pretty fast, but the deep STI gives a more stable performance. We further tested this using for in vivo tractography using data acquired at 70 at 1 millimeter resolution. Here, these data were acquired at 4 head orientations. So as compared to the diffusion measure, we can see that the STI-based reconstruction usually gives like a shorter or less fiber tract reconstruction, but the deep STI can give a much better reconstruction of the fiber tracts, either for global tracking or for tracking over, for example, the corpus callosum. Okay, and the last results I want to show here is it's an interesting result that when we apply the deep STI in the MS lesions, so there are 2 different MS lesion types. For the QSM hyperintense lesion or the paramagnetic rim lesions, we see a good reconstruction of high magnetic susceptibility or high MMS in these lesions. And interestingly, we also see the neural network kind of predict a decreased susceptibility anisotropy, which is related to possibly demyelination. And for the QSM iso intense lesions, we know from the Chi separation results, these lesions are likely to have both iron and myelin loss. And as the deep STI results shows, we have kind of iso-intense MMS, but we have a kind of slightly decreased susceptibility anisotropy. So this all kind of agrees with what we have seen before in the Chi separation results. So as a summary, I think I've shown you that the STI can better describe the anisotropic magnetic susceptibility in white matter. It can be used for MRI histological tools for ex vivo studies, especially when we can push to the high resolution. And it may have a potential for high resolution imaging of the tissue anisotropy in vivo if we have a fast acquisition and better regularization approach, such as shown in the deep STI work. So with that, I would like to thank all the colleagues I've worked with, both in the Kirby Center and the Johns Hopkins University. And with that, I would like to finish my talk. Thanks for your attention.